everyone, in this video we are going to check the facts about protein synthesis or translation. Protein synthesis is a complex process includes many events which are coordinated by translation proteins called as initiation and elongation factors, which make sure each and every step is brought into place while protein synthesis. Protein synthesis is a process of incorporating amino acids to build the polypeptide chain and protein, following the genetic codon sequences arranged on mRNA. It's the genetic codon sequences or alignment which is determining the amino acid sequence or composition of the protein. Discussing about the protein synthesis, the union of all three RNA molecules, those are tRNA, mRNA, rRNA, is an compulsory event. As you can see in this picture, the ribosomal RNA is associated with the mRNA to start the protein synthesis. The ribosome, by moving along the mRNA, reads the genetic codons of mRNA and makes sure the amino acids are arranged according to the genetic codons arranged on mRNA. So first, as we can see, the important point to be noted here is the mRNA site of attachment is falling between the small and large subunits of ribosomal RNA. So how to fix this? So in order to make this association possible, first we need to separate the ribosomal subunits with the help of initiation factor 1 and 3, which binds to smaller subunit and keeps the small subunit away from the large one. The initiation factor 6 binds to the large subunit, performs the same. Simultaneously, the initiation factor 2 joining with the GTP combines with the initiation tRNA, which is bringing the methionine against to the initiation codon AUG, forms ternary complex. Now the ternary complex binds to the 40S smaller subunit in eukaryotes or 30S smaller subunits in prokaryotes to form as pre-initiation complex. Now the pre-initiation complex start to recognize the 5' end of the mRNA and it moves along the leader sequence of the mRNA at the 5' region until it finds the initiation codon AUG, and there it will stop. So after this, the initiation factor 5 hydrolyzes the GTP attached to the initiation factor to utilize the energy released from the GTP hydrolysis for the correct placement of tRNA on AUG or against to the AUG initiation codon. Simultaneously, the energy is also used to remove the initiation factor 1, 3 from the smaller subunit, so to favor the reassociation of the smaller and larger subunit. Therefore, the 70S initiation complex in prokaryotes or 80S initiation complex in eukaryotes have been formed. The larger subunit is divided into three portions mainly, which are A site, P site, E site. So every time the ribosome makes a movement along the mRNA from one genetic codon to the another genetic codon, the tRNA will be moving from A site to the P site and the P site tRNA moves from P site to the E site respectively in order to facilitate the polypeptide chain synthesis while ribosome is starting to read one codon to the another codon. The first tRNA, which is bringing the methionine, is always fixed on the P site so that it will welcome the second tRNA to the A site and associates with it by peptide bond. The amino acid of P site tRNA molecule will join by peptide bond with the amino acid of the tRNA molecule in the A site. This peptide bond formation is catalyzed by peptidyl transferase. Peptide bond is the bonding between the amino acids hanging onto the tRNA of P site as well as A site. After this, the P site tRNA molecule hands over the amino acid to the tRNA. So the chain synthesis started taking place. Now the initiation complex or the initiation phase starts to move into the elongation phase. For the elongation to take place smoothly, the two factors play very important role. Those are elongation factor 1, which brings the new tRNA molecule to the A site having GTP on it, and the second factor is elongation factor 2, which helps the tRNA molecule to make a movement from the A site to the P site and P site to the E site during elongation phase. Now the amino acid chain is synthesized and the ribosomes are kept moving until the stop codon is reached. As the stop codon does not have complementary tRNA molecule, the polypeptide chain synthesis will be terminating at the stop codon. As soon as stop codon appears in the A site, the releasing factor binds to the A site and helps to dissociate the polypeptide chain from the tRNA molecule in P site. So the released polypeptide chain may enter into the cytoplasm or into the endoplasmic reticulum for the post-translational modifications. Is describing the various events taking place before the formation of initiation complex. First, the larger and smaller subunits are separated via the initiation factor 1 and 3 binding to the smaller subunit, and the initiation factor 1 and 3 keeps the This picture depicts what are the various events taking place before the initiation complex formation. In this picture we can see the separation of the smaller and larger subunits of ribosomal RNA by binding to the initiation factor 1 and 3. The smaller subunit is dissociating from the larger subunit and it is ready to mingle with the ternary complex comprising tRNA molecule initiation factor 2 attached to the GTP. Now this complex is ready to join with the mRNA molecule. mRNA they join by recognizing the leader sequence present at the 5' end of the mRNA. This complex identifies the leader sequence and starts to move along the mRNA by making a contact with the mRNA. It will move until it finds the first initiation codon AUG. This is called as pre-initiation complex having a smaller subunit attached to the initiation factor 1, 3 and initiation factor 2 attached to the GTP and methionine on it. And now the pre-initiation complex joins with the larger subunit back again with the help of initiation factor 5, which hydrolyzes the GTP attached to the initiation factor 2, and the released energy is used for bringing the larger subunit towards the pre-initiation complex and makes the larger initiation complex, that is 70S initiation complex in prokaryotes and 80S initiation complex in eukaryotes. So the initiation factor 5 hydrolyzes the GTP. The energy released from the hydrolysis of GTP is used for bringing back or reassociate the larger subunit with the pre-initiation complex and join to form larger initiation complex, that is 70S in prokaryotes and 80S in the eukaryotes.